Okay, good afternoon, grade 8.
Good
We are
afternoon, sir.
in the middle of the discussion and we need to hurry up. I'm sorry if I cannot give you some of the examples or accurate or specific examples simply because we're running out of time. Out of time. Your exam is fast approaching. That would be this April 16, 2021 at 8 a.m. Correct? Right.
Yes, sir.
Let's continue. Cell cycle. Eukaryotic cells, Yo means two, karyon means nucleus. So these are cells with a two nucleus. Undergo an orderly set of events which culminates in the production of new cells. This set of events is called the cell cycle. Okay? Let's tackle more, dig more about cell cycle. It is divided into four distinct phases. Take note, phases of cell cycle. Okay, we have G1, S, G2, and the M phase or mitotic phase, right? Let's move forward. The control of the cell cycle is influenced by both external and internal factors, nutrients and growth factors, as well as the size of the cell, as well as the cell size may trigger a cell to divide. Cell cycle is a complex process involving special proteins that control the switching on and off of these four phases, right? These points or these control points assure that the cell cycle is progressing as it should, right? CDK or the cyclin dependent kinase or kinase, this is a this is one of the major control switches that signal the cell to proceed to G1 to S or from G2 to M phase. Okay. Another substance called P53 is a protein that blocks the cell if the DNA is damaged. This is important protein simply because it inhibits the cell to divide if there is a damaged DNA. Damaged DNA would mean damaged set of genes. Okay? If DNA is too damaged, this may lead to cell death or apoptosis. Okay? It should be removed from the system because the DNA is damaged. And again, this may lead to the damage of the genes also, the set of genes. Recent studies reveal that the P35 is frequently mutated in human cancers. Okay, cancer result when the regulation of cell cycle goes out of control and the normal cell growth and behavior is lost. So again, what will happen if there is a continuous cell division with a damaged DNA? This will result to a damaged also cell or this will result to a cancer. Okay? If you have question, please raise it in our class, right? So this is now the cell cycle. Okay? Again, what are the first one, the CDK or cyclin dependent kinase is one of the major control switches. If there is a switch to continue, there's also a stopper. Okay? The stopper is your P35. Again, if there is a CDK or cyclin dependent kinase means it is a uh, kinase is this is the factor that controls the switches in moving forward. Are you still with me? Grade 8?
Yes, sir.
Yes, sir.
All right. Just give me a minute. I'm just preparing something for you. Again, going back to the discussion, CDK is an enzyme. When you hear the word kinases, these are enzymes that control the switching of the cell to proceed to G1 to S or G2 to M phase. Okay. By the way, if you're going to look at this, G1, S, and G2, these are the preparation stage for cell division because M phase is the mitotic division or mitotic phase. Okay. Now, we will try to look at how this or how the cell prepares itself for cell division. Okay, again, CDK is the one who control the moving forward, and the P53, this is not 35, this is 53, is the protein that blocks the cycle if the DNA is damaged. Okay, this is the blocker or the stopper, and the CDK is the one who let the cell continue the process. Okay, right? So this is the layout of your cell cycle. G1 is growth and normal metabolic roles, the first growth phase. Okay. S is the synthesis phase. This is where the DNA replication take place. And the G2 is the secondary growth or second growth phase. Okay. Growth and preparation for cell division. This is if the DNA is already replicated in the synthesis phase, therefore, the cell will move forward to G2, which is the growth and preparation for cell division. Okay. G1, S, and G2, it is considered as the interphase or the preparation phase, right? Before mitosis. Mitosis is the main event in the cell cycle. Let's take a look at these phases and what are the events, the important events happening in these phases. Okay. This may result in a tumor or clump of cells that grow in it if the cell divide abnormally. Some tumors, though they are harmless, such as fibroid cysts in the uterus, but can become malignant. Okay, we have two phases, or we can consider as cysts, or there are two classifications of cysts. We have benign and malignant. Okay, malignant is considered as a serious damage or serious problem. Okay, benign is harmless. Okay, again, but it can become malignant too, which means that the tumor grows uncontrollably and destroys the surrounding and nearby tissues. And as more tissues are destroyed, it can cause the death of an individual. Cancer cells differ from normal cells in that they divide rapidly. Remember that the cell takes time to divide because it needs to prepare itself for what? For cell division. They tend to wander or more about and invade other tissues. Okay, they tend to invade. Okay, they tend to damage also the DNA. And lastly, they produce different surface proteins as compared to the parent cell it came from. Okay, it produces a lot of proteins. So we need to say, if it produces a lot of proteins, it also produces a lot of damaged cells. Exposure to mutagens or agents that cause genetic changes like X-rays, gamma rays, or carcinogen or cancer-causing substances, just like your soft drinks. Now, your soft drinks is considered as a cancer-causing because of the substance called um, there is a substance that can cause cancer in your soft drinks. Okay, it's a sugar. Right? We consider that as a magic sugar. Okay, activation of the oncogenes. Remember that we are all of the human beings has oncogenes. Onco means a cancer cell, inactivated cancer cells. We are we do have that one, but it is inactivated. Once the oncogenes will be activated. Therefore, you can have a cancer. So the activation of these oncogenes um, may trigger or be triggered by the carcinogens or your cancer causing substances. Oncogenes or genes with the potential to cancer cause or causing cancer or a combination of these have been linked to the cause of cancer. Okay. Even in female, they have this breast cancer. No. There are a lot of factors why breast cancer actually occur. In males also, we have prostate cancer in older men, right? There are a lot of factors also that causes these cancer. One of the cancer causing is the carcinogens. Okay, the vices, those are cancer causing. Okay, do you have any questions so far?
Answer.
Right. The first three phases prepare the cell for cell division by duplicating its genetic material and producing organelles and cytoplasm for the future daughter cells. Okay. The initial phase, we are now um, in the cell cycle. G1 or GAP1 is the first growth phase. During this time, the baby cell grows and prepares itself for DNA replication. Enzymes and protein necessary for DNA replication are also produced during the latter part of G1. Next, after G1, the cell moves now to S phase or the synthesis phase. Okay? This is the time when the cell replicates its DNA. The identical chromosomes appear thin and thread-like as the chromatin fibers that make up the chromosomes are still uncoiled. Okay. As soon as a cell divide will not proceed to the next, or the divide cell will not proceed to the next phase if the DNA has not been completely replicated. Okay. Again, it will not go to the next phase if the DNA is not yet replicated, because the essence of the synthesis phase is to replicate the DNA for possible cell division. Centriole also divides at this point. This is in preparation also for cell division. The G2, after synthesis, after the DNA is being replicated in the synthesis phase or S phase, it will move now to G2 or GAP2. It's the second growth phase in which the cell manufactures the necessary materials it will need when the cell divides, such as organelles, materials for new membranes, and the cytosol. Okay? When these materials are ready, the cell then proceeds to M phase. Okay? M phase or mitotic phase of cell division. Together, G1, S, and G2 make up the interphase stage of mitosis. Meaning to say, this is the preparation stage. Are you still with me? Grade 8?
Yes, sir.
Okay. While some describe interphase as the resting stage, because this is considered as the interphase, the G1S and G2 are considered as the interphase, as the resting stage. The cell during this time is in fact very busy preparing for cell division. This is now the preparation stage for cell division. The M phase or the mitotic phase is the process in which the nucleus divide into two nuclei. Each new nucleus has the same number of chromosomes as the parent cell. A parent cell contains two complete set of chromosomes or diploid or 2N. Okay? Remember what we had discussed last meeting about N and 2N? N is haploid or contains a half number of chromosomes, and 2N is diploid or contains the complete set of chromosomes. It commences with prophase. This is the first phase in mitotic cell division or the M phase, which consists of changes in both nucleus and cytoplasm. The nuclear envelope breaks down, nucleolus disappears, duplicated centrioles begin to move in the opposite poles to form spindle fibers between them. And the chromatin fibers become visible as they condense, coil, and become shorter and thicker. So I will let you see what a cell in prophase would look like under the microscope. Okay, we have specimens and we taken pictures out of it. Oh, it's too long. The duplicated chromosomes held together by a centromere, it is in the center, now appears more distinct, and the two identical chromosomes are sister chromatids. Okay, if you're going to divide the chromosome, you can actually see two sister chromatids begin their journey to the equator. Okay, they attach to the spindle fibers by the way of structures in the centromere called kinetochores in the center. Again, they are still moving towards the equator. Okay, the chromosomes are still moving towards the equator. During metaphase, this is the second phase of the mitotic cell division, chromosomes migrate randomly and line up towards the equator. Another checkpoint occurs at this stage. When the chromosome is not properly aligned at the mitotic spindle, mitosis will be arrested or stopped. Okay? Because mitosis, they need to align, the chromosome should be aligned in the mitotic plate, okay, or the equator itself. When the chromosome is not properly aligned, therefore, mitosis will be arrested or stopped. This is to ensure that the complete set of chromosomes will be distributed to each new nucleus. As a result of this checkpoint, sister chromatids do not separate until a complete set has been organized, ready to be passed on to each daughter cells. Okay? Remember that they need to form a recombinant DNA. They need to pass the genes from one to another. That is the beauty of nature. Okay? That is how we acquire our genetic characteristics from our parents. In anaphase, that's the third phase of cell division or the M phase, mitotic cell division. The sister chromatids that make up the duplicated chromosomes separate and move towards the opposite ends. So they are actually pulled by the spindle fibers, okay, pulled away from each other or opposite to each other. Okay. Once each sister chromatid separate, it is now a distinct chromosome. Okay, that is in anaphase, and the migration is made possible because the proteins that make up the spindle fibers contract and pull the chromosomes apart towards the opposite ends. Okay, they're actually held by the spindle fibers. Sure, what are those? The spindle fibers, the centromere, the centrioles. Okay, let's tackle about that at the end of this cell division because I created an illustration that will let you see the purpose and will let you see what is beyond those organelles. Okay. By the way, centrioles, the spindle fibers is actually connected to the centrioles. Remember, um, in the start of the cell division during prophase, the centrioles move towards the opposite poles. That is in preparation for the pulling of the sister chromatids away from each other. Okay, with the use of the spindle fibers. Last but not the least, the fourth phase of cell division or the mitotic cell division, we have the telophase. Nuclear form in both poles as new nuclear membrane forms around the new nucleus from the remnant of the parent cell and from the membrane materials built during the G2 phase. Remember the G2 phase? It actually prepares the cell for cell division and the materials needed also for cell division. Nucleolus reappears and spindle fibers begin to disappear. This last stage of mitosis is somehow opposite of the prophase. Okay. Diba? When you go to prophase, the spindle, the nucleolus disappears. Okay. And the centrioles move towards the opposite pole. By this time, in telophase, it's different because the spindle fibers disappears and the nucleolus reappears. Okay. Cytokinesis now is happening after telophase. What is cytokinesis? Cytokinesis is the division of the cytoplasm, begins before the mitosis and ends in the late telophase, and is completed when the cell contents have been divided equally to the two daughter cells produced. Okay. Sure. How can we identify if the cell is in cytokinesis? The cell is into cytokinesis if, number one, you can see a furrow or you can see a cleavage or cleave. Right? Are you still with me?
Yes,
Yes,
sir.
sir.
Okay. Again, cytokinesis happens when the telophase, it is in the late telophase. And how can you identify that the cell, by the way, sir, how can you identify that the cell is in telophase? The cell is in telophase if the cell is multi or uh, binucleated. Bi means two. Okay? Two nucleus in one cell. That is now in the telophase. And if the cell undergo cytokinesis, it will form cleavage or cleave or furrow. Okay? That is the time that the cell now tries to separate from each other to form two daughter cells. In animal cells, cytoplasm cleaves or creates a cleavage to form a new cell. In plant cell, however, the cell plate forms at late anaphase between the two new developing nuclei. Centrioles are also absent in plant cells. There's no centrioles in plant cells, but similar structure that does the same function is present, which produce spindle fibers. Still, they they are able to perform cell division because there is a similar structure that actually present in plant cell, which also creates spindle fibers. Mitosis may or not be accompanied by cytokinesis. Yes, resulting in cells that have more than one nucleus or binucleated or two nucleus. It will become multinucleated. Multi means many nucleus. Okay. So this is the illustration of the cells undergoing cell division. Okay, this is G2. This is the preparation stage. Okay. The centrioles now is actually trying to move. Once it separates, it will now perform the prophase. It will go to the different area or opposite ends. Okay. This now enables the cell to undergo prophase. Okay. In metaphase, okay, in metaphase, cells aligned at the equatorial line or metaphase plate or equator. As simple as that. They're just the same. Okay, they're just terminologies. Once they're aligned in the equator, spindle fibers attached to the centromere of the two of the chromosome or two sister chromatids. Okay. Once they're attached to the spindle fibers, it will pull. So the spindle fiber pulls now the sister chromatids away from each other. Okay? And after which, it will go to telophase. Okay, this is now the telophase. This is the cleavage that I'm telling you a while ago, or cleave, or furrow. Okay, to separate two daughter cells. Let's have this for a while. Okay, same as mitosis or mitosis. Mitosis also, if the division is into 2n, 2n means diploid or complete set of chromosomes, it is considered as mitosis. But if the division is n or haploid, number of chromosome, it will undergo meiosis. Okay? If mitosis ensures that each daughter cell will receive the same number of chromosomes as the parent cell, another type of division or cell division ensures that the number of chromosome is maintained among the same species by reducing the chromosome number by half, or haploid, or n, in an organism's sex cells or gametes. Right? Do you have any questions so far before we proceed?
No, sir.
None,
Okay.
sir.
Still have 2 minutes left. This type of cell division is called meiosis. Okay? While mitosis and meiosis share similarities because they are similar, they are different in two aspects. Number one, recombination if genetic materials occur which results to variation. And number two, 4 haploid cells are produced at the end of meiosis. In mitosis, we have two. Okay? We have two daughter cells produced in the end of mitosis. But in the end of meiosis, it's 4 daughter cells. Except for oogenesis. Because in meiosis, we have two categories or types. Okay? In meiosis, we have oogenesis, or the creation of egg, and spermatogenesis, the creation of sperm. Okay? By the way, again, what enables the cell, or what enables the cell to go from G1 to M, or from M to G2? What enzyme is involved? Grade
Is
8.
the what again, sir?
What enzyme is involved in the moving on of the cell from G1 to S, or from S to G2? What enzyme? It is the
The cyclin.
correct, the CDK again. Okay, the cyclin dependent kinase, right? And what inhibits now the cell division or stop the moving on of the cell